So the class will have 4 work to do today, and 3 we do today, 1 will be introduced to you. So what you have is this heavy fact sheets. So what you see at the beginning is called a biochemistry test. This is what we're going to have to do. This lab, I think 11, I call Biochemical test 1. So our lecture after next week, we will move to exam 3, will be biochemistry and genetics. And our lab at the same time, corresponding to that, will do the biochemistry test for bacteria. And we will introduce you around 7 to 10 tests. Okay, so we'll do these one by one. Among them, we will be inserted the clinical microbiology lab, and today we'll do one. So I'll explain to you later why we wanted to do that today. So first of all, I want to give you some of the introduction. We can use biochemistry test to do the identification of bacteria. The reason is that we have substrate. And lots of the time, bacteria has enzyme. And they will generate a products. And the products lots of the time is either acid or gas or alkaline. So basically is this. This is usually bubble. And the last one, hydroxy. And the products in the bacteria biochemistry testing media usually will show you color change or bubbling. Therefore, we know something is happened. So we say it's test positive. Now, how accuracy the biochemistry test to determine a bacteria? Usually 70 to 80% accuracy. The reason is that lots of the bacteria, they will result the similar or same biochemistry results. So we cannot solely rely on biochemistry test. That's the reason we need to do the gram stain. We need to see the morphology of the single colony, and we also need to do the molecular identification, also like a latex agglutination, these immunology determination, plus biochemistry test. Okay, so this is some overview, like introduction. Now the detail of these, let's say what type of the substrate, lots of the time is sugar, carbohydrates. And how they generate products, it's going to go into like glycolysis, fermentation, carbonic acid cycle, all those things we'll talk one by one on the lecture. It's same as your biochemistry class. But here there is one thing. How do you know it is an enzyme? ASE means enzyme. So for example, catalase. For example, urease. Oxidase. All these means it's an enzyme. Okay? So what's the work we're going to do today? We're going to do test 1. Urea test. Okay, here is something I want to let you know first. Biochemistry test you do is very easy. So basically, you get a urea tube. And then you put the bacteria there, it changes the color, then it's positive, it's negative. Oh, that's simple. Sometimes it's a beautiful color, sometimes no color change and negative. But what we need to learn is the story behind that, which means a little bit of mechanism behind it. Why the color is changed, why the color is not changed. That sometimes is a little difficult because there is a lot of chemistry there. Okay, so we want to talk about the chemistry, structures, the principles behind that. And we also have this fact sheet is heavy, so today urea test is on the page 1, which is page 67. And we also going to talk about the fermentation test, which is followed by that. And catalyst test is exercise 3. But catalyst test we're not going to do what we talk in the fact sheets. We're using a different way to do. So urea test. First of all, this is urea. So we need to talk a little bit of structure. This is urea. Urea widely existing in the liver. If the bacteria has urease, which is enzyme, the urea will break down, become ammonium and carbon dioxide. The carbon dioxide usually it's a gas. Okay. This is a magic stuff. Ammonium weak alkaline. And I just said the products, if it's alkaline generated, it's going to change the color. Yes, it can change the color. Because in this tube, we add something there. Add a pH indicator, which is called phenol red. And phenol red, it is very interesting. If I have a pH at 7, pH at 14, and this is usually pH at 1, pH at 14, it is cherry red. And how about pH at 1? And in the acid environment, it is yellow. And the pH 7, it is original color pinkish. So what this means? You are inoculated bacteria there. After incubation, and we'll just put on your tray, okay? We incubate room temperature for 2 days. If you see the color change, become cherry red, then it's positive. So, how are we going to do today's work? Today's work is very simple. Each of you going to do, you will grab a urea test tube, inoculate a bacteria. If you are inoculating E. coli, you bench partner inoculate Citrobacter. Okay? So let's say from the bench number 1, you inoculate the E. coli, you bench partner inoculate Citrobacter. Then the results you share on Thursday. Okay? It's very simple. Now what are we missing? Although you're going to put on your tray, we're not going to mess it up. You still need to do a label here. Bench number, initial, bacteria name, and date. Okay? And this tube, ideally 35 degrees Celsius 24 hours. What we will do, 25 degrees Celsius 48 hours. It's the same thing. Why are we going to do this? Otherwise, you will spend a half an hour to find your tube on Thursday. Going to wasting a bunch of time. We want it to be more efficiency. So everybody put on the rack on your tray and took it out on Thursday to see whether there is a color change. This guy is a risk for some of the bacteria. I do not know whether you have heard about it. Helicobacter pylori. If you look at this one, you will see that. The urea test is very typical for specific bacteria. This is a human pathogen, a severe human pathogen. widely existing in the stomach. Now why? We learned in the lecture, stomach is very acid. The acid environment is pH 1 to 2. But why this bacteria can survive? I tell you, not only survive, it can grow and cause possibly the stomach cancer. The reason is this bacteria has ureas. They can break down urea, become ammonia. This is a weak alkaline. They can buffer lower acid environment of stomach. This pH roughly 1 to 2. Okay, so that's why this pathogen is a risk, because it has ureas. What is this? What is this? That's fat. So, if people has liver disease, what's the first symptoms? The preclinical symptoms is you keep farting all the time. That's gas. And the abnormal level of the urea in the liver, it could cause accumulated amount of the gas generated will become a fat. It's very simple. A part of the reason. Okay, of course there's other gas components. So that's something we talk about the urea test. So like I said, the test is simple. You put a bacteria, you see the color change, but you need to know the principles behind it. Test number 2. This is called 3-tube fermentation test. Okay, what we do, you will get 3 tubes. These 3 tubes containing 3 different types of the carbohydrates, which is glucose, sucrose, and lactose. Glucose, you know about that. Sucrose is not a reducing sugar. It's a disaccharide. That's lactose, a reducing sugar. And the lactose, some of you cannot take it because lactose intolerance, you don't have the enzyme. The enzyme called glycosidase can break down lactose, become glucose and galactose. We're going to talk about this in the lecture. We're not going to draw this structure here. It's too complicated. Okay, so what happened for this fermentation? If we want to say fermentation, very simple. It is like carbohydrates, usually without oxygen, they will generate acid and gas. Okay, how do we know the acid generated? The same thing, it turns yellow. So, 3-tube fermentation test is also very straightforward. Let's say you have a glucose. You inoculate the bacteria, they put the bacteria inside. And then we do the incubation, and then you see the yellow color comes out. It's acid comes out. So that means it's acid comes out. Now how do we know the gas comes out? If you look very carefully, inside there is another tube, a very tiny tube. So every these 3 big tubings and there's a tiny tube. This called Duran tube. This used to test the gas. So how do we know the gas positive? After incubation, if you see the bubble comes out, then gas fermentation possible. Okay, so today's work is also very simple. Everybody gets 3 tubes. King cap tube, be careful, number 1 accident in the lab. Every tube is like this. You have glucose, sucrose, and the lactose. We also inoculate the bacteria there. If you are doing E. coli, you batch partner doing Citrobacter. Again, those tubes, we incubate 25 degrees Celsius 48 hours, which means in your tray. And then on Thursday, we want to see is there a color change. We'll determine positive or negative. And very important, do not forget, label at the top, initial, batch number, bacteria name, and date. So these are the things we're going to do biochemistry test. What else are we going to do? We're going to do the first clinical lab, throat culture. Usually this test we are doing later, but why do it today? We will have a lecture exam 2 next week, Tuesday. Today and Thursday, when you do this, you get some hands-on experiences about Staphylococcus, Streptococcus, or possibly Corynebacteria. Where are the material? The material is chapter 19. And do not forget, go ahead to read. These are very good material, which is concise than what we put in the eCampus after class reading. So, this chapter you should read to prepare the lab, also prepare the lecture exam. Now what we're going to do? So the next one is a throat culture. This is actually lab 12. How are you going to do? 2 of you are going to work together. One is a nurse, another one is a patient. This is already autoclaved cotton swab. You're going to have to wear gloves. I wear one, okay? You have to wear 2. Take out one of the cotton swab. Open your mouth, say ah, and you need to get from tonsil area. And be very careful, needs to be exact tonsil area. It's not your oral cavity, not the teeth. Why? We'll have an exam on Tuesday, so you need to know. If you get it from oral cavity, you will get all kinds of streptococcus. We call Veridyne streptococcus. Remember? Then you get streptococcus, bovis, mutis, multis, mutants, all kinds of things. It's ugly on the blood agar. You will see it's terrible. That's not what we want. So you need to be get from the tonsil area. After you get it, what we do? We have a blood agar. So a blood agar we're gonna do the same. Very intensive using cotton swab to do the first gauge. Change to the loop. That's good. 3 gauges is enough. Okay? And this we will be incubate in a candle jar. So once you do the cotton swab, let's say you have a sample, you're gonna have to do strict plating onto here. Okay, this is already contaminated, so it doesn't matter. I'll just do a demo. Very intensive at the beginning. This is already contaminated, so it's okay. I'll just do a demo. Very intensive, okay? Then, using loop to do second gauge. You change to the loop to do second gauge, and then do the third gauge. And then, flip it over. Put it here. Candle will be flaming and put on the top of the jar. Why the candle is enough? Why the candle is important? Did you ever go to the cave? How do you determine there's oxygen there? You have your candle or some of the fire there. When you walk in, the fire is gone, the oxygen is gone. The candle jar will create microaerophilic environment. Basically 5% oxygen, 10 to 12% carbon dioxide, and the left nitrogen. This is around 75% nitrogen. Why we need microaerophilic environments? Because we could have created beta hemolytic colony. The beta hemolytic colony or alpha hemolytic colony, that will tell you it could be staphylococcus or streptococcus. And lots of these colonies we call pinpoint colony. And I guarantee you on Thursday, some of the students in this lab, you will find in your own throat culture, you have staphylococcus and the streptococcus. Okay, that's the work we're going to do today. And on Thursday, once you get this agar, blood agar, we will first of all look at the colony. Then we go ahead to do the catalase test. So why we do catalase test? Because catalase test can differentiate staphylococcus and streptococcus. Is that right? Staphylococcus is positive, streptococcus is negative. So from that point, we have one way or the other. And then we do the gram stain, you will see a beautiful morphology and arrangement, and that's from your own culture. That's the reason why I wanted to do it today, because it certainly helps you to review exam 2 and get hands-on clinical experiences. So that's why I want to do it today. I used to be moved very late, like in November, but I think you have an exam to prepare, and we're going to do it today, then you get these hands-on experiences. Okay? That's very good. So, how about that? I'm still going to do a demonstration to you. Because although it's very easy, but I would still would love to do a demonstration for these tests. First of all, when you came here, okay, make sure you do in order urea, so everybody came here. We'll do the demonstration. I know it's simple, but I still wanted to do it. Because I would love everybody to get it. Otherwise, you're very confusing what it looks like. Okay? First of all, using your personal tray, go there to get these tubes. You're going to have an order like this. Okay, of course wear gloves. Do not use your bare hands. That's 4 tubes. If you remember, that's good. If you don't remember, please write on your notebook. What are these? It's very important. You're going to mess it up. What is this? Urea tube. Looks rose color pink. What is this guy? Glucose. What is this? Sucrose. What's that? Lactose. How do we know it? The color of the cap. So you can write down the yellow color is glucose, blue color is lactose, and the white color is, the second one is sucrose, and the white color is lactose. We have the label on the rack already, so you can go there to do it. Okay? That's very important. Do not mess it up. Okay, what we do next? You're gonna label on the top. Okay, I'm gonna skip that one. Label on the top. And label on the top. And label on the top. Now how do we do it? Just a very quick do a demonstration. I think I need to do it because although I think it's easy, some of you still confusing. This is good. Do you have gas today? No gas?
No, we have gas.
Hockey stick. Good, now. Terrible.
Yeah, use this one.
No, man. That's not very scientific, okay? That's not very, don't do this, okay? Don't do this all the time. Occasionally is fine. Flame the loop. Okay, simple. Only one. If you do E. coli, you batch partner do citrobacter, not do the double. Same thing. Little finger, flame, touch the bacteria, flame. Assuming you labeled flame. That's all. Okay. Then when you finish, put it here. Okay. Second, just give an example. One more example. Okay. Flame the loop again, cause different reagent. What we do? Let's say E. coli. Flame the loop. Get the bacteria. Flame the loop again. Go here. Be careful. That's all. Don't block the Durant tube. Okay, that's important for testing the gas. Otherwise, you're not going to see it. That's all. So finish all of these. Very good. Be careful.